Again, the important unit for the botany minor examination is virus. The first aspects about the virus is who discovered the virus. The answer is D. and Walsh discovered the virus. He explained that infectious agent smaller than the bacteria can cause the disease. As that infectious agent that causing the disease is tobacco mosaic virus. Another scientist regarding the virus discovery is Weserneck, that discovered the some agents passing through the bacterial filter and cause the disease. So both are important for the discovery of the virus. Again, what are the characteristics of the viruses? This one is the important question. So viruses, first character is intracellular, that means always found within the cell. Intracellular, obligate, that means their nature cannot be changed, that means always behaves as, that is the parasite. So one of the most distinguished feature about the virus is that viruses are intracellular obligate parasites. Another feature of the viruses are chemically nucleoproteins or nucleoproteinous, that means that made up of nucleic acid and proteins. But the unique about that is the nucleic acid either DNA or RNA, never both. Other than the viruses, both RNA and DNA are present in organisms, either prokaryotes or eukaryotes. But in case of viruses, nucleic acid, either DNA or RNA present never both. If DNA present, that is called DNA viruses, and if RNA present as the nucleic acid, that is called RNA viruses. And this one is the code, like this is the RNA, or this one is the protein. Nucleic acid, this one is the protein. So protein always as a protein as a code, that means that is the outer covering. Some viruses may be capsulated, that means enveloped viruses are also present. Third one, the feature about the viruses are viruses behaves as a living, or that is really multiply within host only. Outside host, that is the viruses becomes inactive in the sense that it really becomes dormant, cannot multiply outside the host. That means for the replication, almost machinery enzymes are used by the host. That means that depends on the host for their metabolism. Again, that will be viruses can be crystallized. This is the non-living property, can be crystallized. अगर आप क्या करो, टोबैको मोजाइक डिजीज का लीफ को डिटैच करो, उसका पेस्ट बना लो, क्रिस्टल बना लो, अगेन डिस्टिल वाटर में डिजॉल्व कर दो और स्प्रे कर दो हेल्दी प्लांट पर, कैन कॉज द डिजीज. दैट मीन्स viruses can be crystallized. This is the non-living property. That means some are living, some are non-living property. So before that, this era, viruses can be considered as a connecting link between living and non-living. But in the present scenario, the viruses considered as a conditional living organism, not as a non-living. Okay, only that the viruses become dormant outside the host, that cannot be dead. That means viruses are immortal. Viruses are immortal, another feature of the viruses. In the present scenario, viruses maybe contains some enzymes like the reverse transcriptase are present in some amount in the some viruses. So viruses are metabolically also active. Also, viruses multiply rapidly. This one is the most distinguished feature as a living character. Again, that is the DNA viruses and RNA viruses that depends on the nucleic acid present in that viruses. और आपको जो पढ़ना है डीएनए वायरसेस में, वो डीएनए वायरसेस में वो किसका पढ़ना है, तो वो है आपका बैक्टीरियोफाज. दिस वन इज द बैक्टीरियोफाज. और एक जो है पढ़ना सबसे जो इम्पोर्टेंट होता है वो T4 बैक्टीरियोफाज होता है. T4 बैक्टीरियोफाज, अगर उसकी हम लोग स्ट्रक्चर की बात करें, स्ट्रक्चर बल्कि यही आवेगा. दिस वन इज द हेड, दिस वन इज द neck of T. Again, this is the head, which is made up of the protein coat. प्रोटीन्स सबयूनिट्स का ये बना होता है. और इसी के सेंटर में ये क्या होता है, डबल स्टैंडर्ड डीएनए होता है. फर्स्ट डीएनए दैट इज द डबल स्टैंडर्ड. दिस वन इज देयर नेक, और दिस वन इज देयर टेल, और दिस वन इज टेल fibers also present. Tail fibers are also present. This one is the structure of the bacteriophages, and these bacteriophages are very common in Ganga's water. That's why the Ganga's water are free from the bacteria due to presence of the bacteriophages. These are the bacterial viruses. Consider the bacterial viruses. So the genetic material in the bacteriophages is the double stranded DNA, and the host of the bacteriophage is E. coli. This is the E. coli. Bacteria that really produces bacteriophages multiply by two methods. One is the lytic and another one is lysogenic cycle. There are two cycles common. In the lytic means in which bacterial cell become their death. That bacterial cell rupture during the reproduction. And in the lysogenic cycle, the bacteriophages multiply without the rupturing of the E. coli. That means in the bacteria. Like this one is the bacteria, and bacteriophages multiply. And once time after many years, that will be bacteria rupture, and that will be releases fast protein. और जब बैक्टीरिया मल्टीप्लिकेशन के साथ साथ अगर बैक्टीरियोफाज भी मल्टीप्लाई करता है, देन दैट इज कॉल्ड लाइसोजेन. और ये क्या हो गया, लैक्टिक साइकिल. सो देयर आर डिफरेंट 5 स्टेजेस. लाइक दैट इज द अटैचमेंट. Like this is the attachment of the bacteriophage, entry into the bacteriophage DNA, not the protein. Only DNA enter into the bacterial cell. And this DNA multiply, reproduce, and produces the methylene. Similarly, another virus important for the examination is the RNA viruses, that is the TMV. that is the TMV. ये क्या है? RNA virus है. This one is the RNA. The RNA single stranded RNA. This one is the surrounded by the protein subunit. Protein subunit that is called capsomere. Capsomere. This is the protein code. Protein code. And number of capsomere is more than the 1000. And this one is the RNA single stranded that causes tobacco mosaic disease in the tobacco plant. That will be DNA enter into the cells of the tobacco plant and multiply, and then translated to form the capsomere. And after assemble the capsomere and RNA molecule, that becomes new tobacco mosaic virus. So this is the all about the unit of the virus. Okay.